On this slide, I have why is the prophage or the virus, provirus stage important. And I think I just explained that at the end of the last one. But so here's the bacterial chromosome, and then the viral DNA gets stuck in there. This can actually code for a protein which makes the bacterium that it's in, so the whole host, so the bacteria, it makes it, wait a minute, it codes for a protein which is a toxin. Okay, so now here's some examples of how all the steps work. So in a DNA virus, okay, so this is going to work just sort of like the HIV video, but so here's absorption, right? So there's step number one. So the virus is in a capsid. The virus is, of course, the capsid and the DNA are together, right? And that's making the virion, and then it's in an envelope. So the envelope will come up and it will adsorb to the plasma membrane of the host of the animal cell. So it's endocytosis. So then the virus will uncoat, okay, and the DNA can enter, let me change the color. The DNA can enter the nucleus, okay, and then the DNA will be transcribed, okay, and then we'll make the virus particles. Okay, translation will occur, okay, and we'll make the proteins that we need. Okay, so we're making the DNA, we're making the proteins, and then when we put them together, we get the virus particles. Okay, and then the DNA can be, or the virus can be released. Okay, I'm kind of babbling around. Okay, so we have absorption, penetration, uncoating. Okay, here's the biosynthesis part. Okay, where we're making the viral DNA and we're making viral protein, and then they mature and they put them back together. They put them together, right? And then they can be released. Okay, now retroviruses are contain reverse transcriptase. Okay, so the retroviruses, what's going to happen is the RNA is going to come in, right? And we're going to form a complementary strand of DNA using reverse transcriptase. Okay, then we'll form double-stranded DNA, and that gets inserted into the host chromosome, so then it hangs out in the host for forever, potentially. Okay, and then one strand of the DNA is going to be the template to make the proteins. Okay, and then we'll put the virus together and it'll get released. Okay, now how do we culture viruses? So viruses, if they have to have a host, how do we actually grow them so that we can study them? So what you have to have, so I have, why is it challenging to grow viruses? It's because you need a host. Okay, so you need a host in order to grow them. So we need a host because they don't have their own ribosomes, they don't have their own, any of their own cell organelles. So we have to have a host in order for them to make more of them. So they use eggs. Okay, so eggs are some of the first things that we use. Eggs are actually used to make flu vaccines. So if you go to get your flu vaccine, some of them will say, are you allergic to ova albumin, which is egg protein, or are you allergic to eggs? Have you ever had a reaction to a flu vaccine before? So the eggs are really, you can stick the virus in there and you can get it to multiply really easily. Now you wouldn't do it by hand, now it would be automated. So it would be a whole big flat of eggs and a robot would come down and kind of puncture the eggs with the needle and put the virus into them. Then you can harvest the virus from it and make the vaccine that way. We also use other cells, so we can use animal cells too. Okay, now virus-like agents. So there are some things that are even smaller than a virus. And the first one we'll talk about is called a prion. Some people say prion. What it is, these are infectious proteins. So they're really small. There's no DNA. So no nucleic acids with them. So this is supposed to be a ribbon diagram, and ribbon diagrams are used to show what proteins look like. So these are two pictures of what a prion would look like. So they're a tenth the size of a virus. They're really, really hard to kill. So they're really, really hard to get rid of. So they're resistant to heat, so you can't autoclave them. They're resistant to radiation, and they don't cause you to have an immune response. But what they infect is they'll infect nervous tissue. So they'll infect neurons. So they cause problems with brains. Okay? And they can be there for a while before you start to have symptoms. They're hard to detect. There aren't any really good detection systems for them. You'd think because it is a protein that you could use an enzyme called a protease, which breaks down proteins to destroy them. But because of the way that they're folded, they're hard to break down. So they're tricky and they're kind of scary. Okay, some examples of prions that are out there. The first time they were found was when they found mad cow disease. So mad cow is also known as bovine, so cow, spongiform encephalopathy. So basically it turns the cow brains into Swiss cheese. 1986 is when it was first discovered. It was in England, so it's in the UK. It's where they found it. They found it because the cows were actually eating other cow parts. So after the cows were butchered, the nervous system, so like the spinal cords and the brains, were recycled into the feed, and the cows were eating that, so it was spreading that way. The cows, their symptoms are they can overreact. Okay, so they're like jumpy, twitchy. They can't walk right. They have an abnormal gait, and they can become aggressive. So they have mad cow. Okay, so it's like, and there's nothing you can do. There's no treatment. Scrapie is another example. It's been around for a really, really long time, and we've been aware of it for a long time, but it just seems to affect sheep and goats. Okay, they kind of lose their coordination and they're itchy, so they scrape. So that's where scrapie came from. Mad cow was bad because it does jump to humans, and that's what they call Creutzfeldt-Jakob disease. So it jumped to humans, and that's what made mad cow especially bad. So if you ate infected meat, then you could get it. So that's why mad cow is so bad. So now they screen like crazy to make sure that mad cow isn't in an area. Another disease that's kind of relatively new is chronic wasting disease, and it's found in elk and deer. And we have it in Maryland, and it's all around Maryland. And the deer look pretty rough. The deer have neurological problems, and their fur is really rough, and they're real thin, so they look bad. I wouldn't eat it if you find a deer like that. There are rules about like butchering deer and moving them from county or hunting region to hunting region. They don't want you to move up the brain and the spinal cord to another area because they don't want the disease to spread that way. There's actually been one study where because they're trying to see if chronic wasting will spread like the mad cow, and they actually fed some primates infected meat, and I think 3 out of the 5 primates got it, but the study was really small. It was in Canada. So it's around. You just need to be aware of it. There are some tests and some hunting areas that they screen for it. Okay, so here I have Creutzfeldt-Jakob again. So Creutzfeldt-Jakob, this is the human mad cow. Okay, so beef definitely comes from, they suspect it can come from pork. There have also been some cases of prions infecting the brain that people have eaten wild animal brains. So there's a report of a festival. It was in the Midwest, and it was all about squirrels, and they ate a squirrel brain stew, which is called burgoo, and the squirrel brain stew got Creutzfeldt-Jakob disease. Ugh. Okay, kuru was also found in humans, and it's been eliminated, but there was a tribe, I want to say it was in New Guinea, but the people, when someone died, okay, somebody died, they ate, that's supposed to be brain, they ate the brain, so they were spreading the disease that way. It was supposed to be an honor that they ate your brain because they're kind of like continuing on your knowledge. That's how they passed on the knowledge. But if somebody had a prion, then they just spread the prion. But it's been eliminated with education, so they don't do that anymore. Brain stew, the people that had... Okay, now viruses and cancer. Okay, so there's definitely a relationship between viruses and cancer. So how they can cause it, there's a couple different ways. So one of the things that it can do, our body has genes that suppress tumors, okay? They keep, if there's a tumor, it keeps the cells from multiplying uncontrollably. Okay, so when you think about like a viral DNA getting inserted into a host DNA, and this is the gene for tumor suppression, if this gets inserted there, it could disrupt that gene. So that can happen. Another thing that could happen is it could actually activate cancer genes, which are called proto-oncogenes. So on this picture, I have a host chromosome. So this is papillomavirus. So HPV is connected to a lot of different cancers. Most of them are like reproductive cancers, so cervical is the big one. Okay, so if the papillomavirus DNA is inserted into the host chromosome at the wrong place, okay, it can cause uncontrolled cell division. Okay, and then there'll be excess viral replication too. So it's just kind of leads to a totally bad scene, totally bad situation. Okay, Epstein-Barr virus actually can cause throat cancer. HBV is hepatitis B virus, and that has been known to cause liver cancer, that's associated with liver cancer. Kaposi's sarcoma, it's becoming more and more rare, but it's associated with HIV. So in the old days, when someone had HIV and it progressed to AIDS, one of the first symptoms of having AIDS would be this Kaposi's sarcoma. So it's a little known cancer, and it only usually occurs in HIV patients. But nowadays, HIV is detected early enough that they don't progress to that point, so that people don't progress to AIDS. They just maintain HIV. Okay, so I think that's it. That's the last slide.